So the next person is John Skidmore. He's the Chief Scientific Officer of the ALBORADA Drug Discovery Institute.
Thanks very much. So while we're getting the slides up, so I'm Chief Scientific Officer of the ALBORADA Drug Discovery Institute, which is based at the clinical school. So we're in the same building as MENA. And our mission is to take the latest dementia science and turn it into new drug discovery projects. And really what we're trying to do is to develop the molecules that are going to be the seeds for the next generation of medicine, and to do so in a way that's differentiated from the work that's going on in industry. So essentially we collaborate with academics in Cambridge and beyond. We work with them on target validation, but our core activity is this cycle of drug design, synthesis and screening to make compounds to test in animal models. We also like to do this in partnership with industry, and this diagram here suggests a rather linear approach. Actually, we like to build three-way collaborations where we can. So here's some examples of some of the things we've achieved over the past few years. We have extensive high-throughput screening capabilities, so we've now run 9 high-throughput screens. And from these, we've actually now identified tool compounds for validation against 7 different biological targets. And for 4 of those, we've got crystal structures showing our molecules binding. We've progressed 4 of those projects into in vivo testing for validation. And currently, we are collaborating with 15 different academic groups across Cambridge, 5 different groups within the DRI, 3 in Cambridge and 2 beyond. And we have 12 other academic collaborations. So we're very collaborative. Similarly, with industry, we've got a number of different industrial partnerships ongoing. And we've been quite successful at bringing in additional funding. So our core funding comes from Alzheimer's Research UK with support from the Alborada Trust, but we've been able to add to this from a number of different funding organisations and also leveraged industrial funding. Key to what we do is to build partnerships, and so those partnerships can be with industry. We have examples here of an autophagy consortium that we ran with a number of pharma companies through the Milner Therapeutics Institute. We collaborate with Sanofi through their Eye Award scheme, and we have a partnership with ADDI Life Science on a particular drug discovery project. Our academic collaborations are very much global, and another key part for us is interacting with not-for-profit organisations. So that might be the DRI, who we've just heard from, and the BRC that we're going to hear from, but also our sister institutes at Oxford and the UCL, and the Rainwater Charitable Foundation, who work with us for us to provide drug discovery capabilities for a number of projects under their TAU consortium. So a little bit about the kind of science that we can deliver. Chemistry is really, really important to us. Here's an example of 4 papers on the PI5P4 kinases that we worked on, where we've really got some of the best tool compounds that are available anywhere for those targets. And we're working on a wide range of different approaches, protein-protein interactions, peptides and peptidomimetics, receptors, covalent approaches, glues and protacs. So whatever your protein of interest, I think there's something that we can hopefully do to work with you. And when we work on a project, what we essentially do is to take multiple approaches to finding hit series. This is an example of a project where we looked at high-throughput screen, virtual screening, scaffold hopping, ligand-based approaches, even doing chemistry and screening direct reaction mixtures to find multiple hit series. We then were able to find an in vivo tool compound which has got good potency and the kind of properties that will mean it will get into the brain. And then once we move into animals, we're very keen not to just jump straight into disease models. So we look at what we call target engagement. So in this case, we have an animal model where we see the change in expression of a gene, which can then be reversed with treatment with the compound. And so we're really directly showing that we're engaging with our target and our mechanism. And this project's now progressed, and lead compounds are being profiled in disease models, and we're working with an industry partner towards a candidate molecule. So that's my final science slide. This is the group who do all the work, and thank you for your attention.